This is a recap of what we talked about last week. We talked about all the different levels. We looked at the organism, we looked at the organ system, down to the organ. We've looked at the tissue level, down to the cell level, and what we're going to be doing today is we're going to be looking at the atomic level, which I'm not going to spend too much time on, and we're going to spend time on the molecular level. So we're going to be going over some of the main molecules that we're going to be seeing in physiology. As we go through our lectures, you'll be able to see the references that we're going to be making. So this stuff is going to build on top of what we're going, this is going to be the foundation of the stuff that we're going to be learning when we look at some of the molecules. This is going to serve as utmost importance because we're going to be learning about things like lipids and how fats interact with the cell that you're going to learn next week. You're going to learn about how water-soluble molecules interact with a cell. Again, something we're going to learn next week. So it's prudent for you guys to understand the makeup of these molecules and the properties of these molecules before we get into physiology. You've seen this stuff before. This is all chemistry. In fact, this is biochemistry. So at some point in your academic careers, you've taken biochem. But I'm not going to delve in as deep as what you guys have learned. I'm really going to cover the basics. And again, some of the stuff you're not going to be seeing after quiz 1, but I will point out the things that will be important for you guys as we go into our later lectures. So our topics for the day. We're going to look at our atoms. We're going to look at when we put the atoms together. We're going to be looking at our molecules. And we'll be looking at the bonds that hold these molecules together. And then when we look at our molecules, we'll be taking a look at two types of molecules, inorganic molecules and organic molecules. And again, I'll talk more about the differences between those. And the main types of macromolecules we'll be talking about that are organic molecules, we'll be looking at DNA, protein, carbohydrates, and fats. And we're going to visit all of those today so that you understand the structure of those. And what we'll be doing next week, we'll be talking specifically about the cell and how the cell is made up with these types of molecules, and at least they'll give you the cellular biology portion. Next Thursday, we talk about the functions in the cells, mainly cellular respiration. How do we generate energy? And again, that will be the foundation before we start to kick into the more complex cells, the cell differentiation, the term that I talked about on Tuesday that refers to the specialization of cells. So now that you kind of have a foundation of what we'll talk about, so atomic level organization. So the structures of your atoms, you've seen this before, it's just a revisit, but we're going to be looking at the contents of the atoms, which we know there's three main components that are typically discussed in at least an introductory chemistry course. Protons, electrons, and neutrons. These are the three main components. I'll talk about those on the next slide. But specifically, when we look at the elements, we know that there's 92 naturally occurring elements on Earth. That's present specifically on Earth. You might think, well, I've seen a periodical table, and it definitely looks like there's more than 92 natural occurring elements. And something we'll take a look at here. A good section of these are man-made. In fact, there are more that are being produced. Things up over here I can't even really pronounce. But these are manufactured in a laboratory. It's stable for a little bit amount of time, but it doesn't exist naturally here on Earth. So if you were to look at the major elements, remember what I mentioned, the ones that we talked about on Tuesday. There were 6 main elements that are found in the human body that comprise 98% of all elements found. And if you remember, that was PCHONs. That was the acronym I had you use. But if you were going to look at 95%, and I know I'm just kind of playing around with the percentages here, really the major elements would be carbon, oxygen, hydrogen, and nitrogen. And when we look at our complex molecules later on today, I bet you that most of that will be this, CHON. The other elements, when we look at the other 5%, some of them will include the lesser elements. Things like calcium, phosphorus, potassium, sodium, chlorine, magnesium, iron. Iron you would have seen in your red blood cells, specifically a protein called hemoglobin that we'll talk about later on that will hold on to iron. These are also required to be found in our human body, but in lesser percentages. And then trace elements. Some trace elements you're going to be able to see. We don't talk too much about them, but we will talk about an element called iodine, something that's required, something that you've seen as iodized salt. It's our intake that we'll talk about. makes a profound influence when we look at our endocrine lecture when we talk about thyroid diseases. All right, so with that, that is our atomic level of organization. If we take a look at, you'll see that the protons and neutrons are clustered around the center, and then we have electrons that are floating around in space around the nucleus of that atom. So then, that again, you've seen before. If you were to weigh these things out, you would see that protons and neutrons weigh about 1 Dalton, otherwise known as 1 atomic mass unit. The electron has actually a negligible weight. It's about like 1 out of 2,000 of a Dalton. So if you were to look at an element, you would see most of that is clustered. Most of the mass of that atom is clustered around the center. And then you've got all these negligible weight electrons that are literally just floating around space, just circulating around. And if you look at the protons have a positive charge, and the electrons have a negative charge. And again, they circle around in these orbitals around the center of the atom. So the two numbers you'll see on here, I'm never going to ask you guys about these, so don't worry about these. But atomic number is the number of protons in an atom. And the number of protons governs what type of element it is. If you were to look at the mass number is the sum of protons and neutrons. You might think, well, mass number, mass doesn't relate to weight. Well, we know that protons and neutrons are about 1 Dalton each. So if you count the number of protons and neutrons, that tells you exactly what your mass number is. And you'll be able to see this in our periodical table of elements in just a bit. Actually, you can even see it here. So if we look at the atomic structure, we're going to look at CHON, and this is hydrogen, carbon, nitrogen, and oxygen. These are the 4 main ones we're going to talk about. We'll talk about sodium and chlorine, maybe not so much potassium and iodine, but at least the idea I want to get across is, again, you'll see the protons in the center, neutrons in the center, and you'll see the electron floating around. One of the key things I want to point out, again from basic chemistry, electrons float around the center of that atom, but they travel around these orbital shells. And again, I'm not going to get into too much detail about it, but the general rule is that the first shell, and you'll see these shells that are surrounding each of those proton-neutron cores. The first shell holds about 2 electrons. The second shell, and every shell thereafter, third to fourth, etc., will carry about 8 electrons each. And I know there's other orbital shells, so if you're a chemistry guru here, don't knock me on my chemistry. I'm here to teach you guys physiology. But the main idea behind it is, if you take a look at, let me go into carbon. You'll see carbon has 6 protons. That's just a fact. If you were to count the number of protons, carbon, it's always 6. If you look at the number of neutrons, in this case, we have 6 neutrons. So 6 plus 6, 6 Daltons plus 6 Daltons, for that of proteins and neutrons, give us a mass number of 12. Okay, I'll talk about this 13 in just a little bit. If we take a look at the electrons, you'll see that we have 6 electrons floating around the shell. So knowing that we have 2 electrons in the first shell, that means if there's 6 electrons, that should mean there should be how many in the outer shell? 2 electrons on the first, 4 on the outer shell. Okay? So I'll talk about that in a little bit when we talk about our carbon atom. Let's give you another example. Let's take a look at sodium. Okay? Sodium has 11 protons. Therefore, sodium should also have electron. If sodium's neutral, again, the topic I'll talk about in a bit, if sodium is neutral, it should have 11 electrons. So given that there's 2 electrons in the first shell, 8 electrons in the second shell, how many are there in the third shell? 1. Okay, so keep that in mind. We have 1 electron in the outer shell all by itself. Take a look at chlorine. Chlorine, if you were to look at 17 protons, you don't really need to do the math. You could really just look at the outer shell. It's already done for you. You can count 7 electrons. Okay? So all it needs is 1 electron on that outer shell, and then it would complete. So now that we have that in place, we know electrons are floating around. We have this general rule here. let's take a look, before I move on to the next slide, something I want to bring up, no, scratch that. We'll move on. The next topic we'll talk about are isotopes. And isotopes, basically, the main definition of isotopes is that atoms of an element have a different number of neutrons. And let me go back to the prior slide, where I pointed out that the mass number for carbon is either 12 or 13. So you're thinking, why is there 13? If the number of protons will never change, right? Because the number of protons in carbon is always going to be 6. If it was 7 protons, well, you wouldn't have carbon anymore, you would have nitrogen. So in carbon, if we have 6 protons, we can see that maybe there are isotopes out there that have a different number of neutrons. And in this case, if there were 13, the mass number was 13, that would mean if there's 6 protons, there's 7 neutrons. And the excess neutrons happen to actually give off a little bit of energy, a little bit of radioactivity that can actually be measured. So when we look at the radioactivity of that particular element, and let me just draw out 13C, the way you would see that notation for a radioactive isotope would be a 13 on there, letting you know the mass number of 13. From there, you would have to look at the periodical table of elements. You would see there's 6 protons. You would know that there's 7 neutrons. So that extra neutron does give off excess gamma rays. There's a small percentage of radioactivity that's given off. And there are applications for it. Okay. So if you have, there's 13C and then there's 14C. This means there's 2 excess neutrons, which actually gives off more radioactivity. And over time, if you had a whole cluster of these, 13C or 14C, radioactive carbon, over time it would start to die off. Because once you keep giving off energy, over time that isotope will turn into something else. What it turns into isn't really the focus of what we're going to talk about, but nonetheless, it still gives off radioactivity. So we've seen lots of applications for this. In fact, the first one I'll talk about is carbon dating. And has anybody here heard of carbon dating? So what exactly is carbon dating? Carbon dating is a way for us to be able to determine the age of a sample. Say a paleontologist finds a fossil, and they take a look at a particular fossil, and they want to find out how old is this? And they say 7 million years. Where do you think they came up with that number? You think they just made it up? No. Actually, what they do is they actually will take a sample of the bone in there, and they know that at any given time, we have a certain percentage of 13 carbon. And I don't remember exactly what that percentage is, but it should be something, let's say, 0.02% naturally occurring. That means if I go and take a look at the carbon, if I were able somehow to grab all the carbon from your body and measure it out, I would know that 0.02% of that would be this radioactive isotope. Okay? Now, if you take that old sample and I measure and I find out that the amount of 13C in there is 0.0005%, I know I've lost a certain amount of 13 carbon. And that's because over time, that radioactive isotope has decayed. Okay? So how long does it take to reduce carbon down to, let's say if I were to take like a pound of 13C, to turn into half that amount? And again, you don't really need to know this, it takes 5,730 years to turn into half of what its original amount is. That term is called the half-life of radioactivity. For a particular element, there's a certain half-life. You guys familiar with the flooding that happened in Japan after the earthquakes? They have all this radioactivity in there, and what they have to do is they have to take all that radioactivity from that nuclear power plant that exploded or whatever it did. They have to take it and they have to store it and wait many years for that plutonium to have its half-life. And it takes a long time for that stuff to decay. They have to contain it and leave it there so that it doesn't affect anybody. And over time, that stuff dissipates and it's no longer radioactive. Chernobyl happened a long time ago. There's still a lot of radioactivity out there, and it's because that stuff is still out there. So they've kind of deemed that area, don't go around because you'll pick up that radioactivity, and that radioactivity can cause things like cancer, can cause damage to your body. So nonetheless, If they were to measure, back to our original thing that we were talking about, if they were to measure that 13C, that 13 carbon, and they found out we had a proportional amount of less, they could figure out the number of years of how old that sample is. Fairly accurately, too. All right, so we're talking about physiology. This isn't really, carbon dating isn't all that important. Maybe from an anthropology perspective, but let's talk about a clinical application. Positron emission topography. This is your PET scan. Has anybody ever had a PET scan here? Nobody? I've had a PET scan done. And what they do is you go into a scanner and they put you in a machine. And it kind of looks like an MRI, but what they do is they inject a certain contrast in you. And this contrast is called fluorodeoxyglucose, FDG. You don't need to know this. FDG is basically a glucose molecule with a tagged, and a glucose molecule is a sugar molecule that you take in, and it has a tagged radioactive isotope on there. So if I were to give this to you, and you had a little contrast, any areas in your body that's taking in that glucose will get lit up. Okay? So what you're seeing here are some images of that. They'll put that person in a scanner, they'll inject them with a contrast, and they'll watch any areas of the brain that's taking in that glucose for energy starts to get lit up. So these are areas that correspond to high metabolic activity. They do these things also with cancer too. They can give you, if they're trying to diagnose and try and find areas in the lung, for instance, that seem to be particularly lit up, like right here. Actually, I don't think this is the lung. But if they were looking at areas that were highly active, they would see, oh, this area is kind of lit up and it shouldn't really be. This warrants some investigation into that organ to see if there are some tumors in that area. So again, FDG is something that they use because it gives off a certain radioactivity, can get picked up by this machine and they can know a lot about it. Something that I did back in college, I know I talked about this on, I think I may have talked about this on Tuesday. I worked in research, but I was also a research guinea pig. And back in college, I was an undergrad. I remember looking at the UCLA newspapers and finding like experiments that they were doing on people. And I would see some that were paying pretty well, and I'd call them up, find out, read the consent form, and it'll tell you exactly what they're doing. And I thought, they're paying $500? Why not? I'll go and start doing these things. And this one thing they wanted me to do was to go in, and don't think I'm a freak just because I signed up for these. I was broke, you know, I needed some money. They had me sit into a machine. Again, they set me up, put me into the machine, into the PET scan, and actually it was an MRI machine, but that doesn't matter. They injected me with the contrast, and they wanted to look at my brain and see the amount of activity that's happening in my brain. Now, they've removed all sensory. They gave me huge earplugs. They gave me goggles so I couldn't really see anything, and they basically told me, don't think about anything. In fact, try and sleep, but don't sleep. And then they want to use that to measure the brain activity. Then they put these goggles on me, and they made me watch this video. And this video consisted of someone, and all I saw was someone picking up a coffee cup and then putting it back down, and it was empty. And I watched this video for an hour and a half. And I remember thinking, they just kept telling me, just focus on the coffee cup, think about somebody picking up the coffee cup and putting it down. And that's all I watched for about an hour and a half. And that whole time they were measuring areas in the brain that would correspond to the whole idea of picking that up. There's a spot in the brain that's specifically attuned to the action of picking something up with your arm. And they found that spot somewhere off onto the side. After I was done with that, the second part of the experiment was they'd sit me down in the chair, they put this huge helmet on, and there was a spike that came on, and they said it was going to just kind of inject a little bit right into the scalp, just a little bit, and they were going to prod that area with electricity. And I didn't do that portion. I'm like, no, you're not doing that. To this day, that happened and I know I'm kind of dating myself, that happened about 15 years ago. I can still see the images in my head. I still see that same coffee cup, and it's not like flashbacks or anything, it's just, I remember this video. Right? Really interesting thing, and I bring this every time I teach it. I found the research article 3 years ago, and I read it, and they had pictures of the coffee cup, and I kind of got the shivers. I was like, ooh, this is really weird. They had like 10 people in this experiment, and it was weird knowing that I was one of those people. So, nonetheless, That's exactly what they did with me. They injected that contrast so they can find that area in the brain that's active. And that's exactly what they do in a clinical setting. All right, so here's our periodical table of elements. Again, something you would have seen in chemistry. But this tells you on here the number of protons. You can see the number of protons for carbon. You'll see 6. And then you'll see the mass number underneath. And again, that mass number is the sum of protons and neutrons, of which you can see carbon has 12.011. So if you're thinking, where's that 0.011? This is an average of all the carbon that's existing out there. And you take an average of it, you'll see that most of it will have 12, at least a mass number of 12. There'll be some that have a mass number of 13, and even a far less number that have 14. The 13 carbon and 14 carbon are the radioactive isotopes. And then they can use that to be able to see inside the body. They can actually tell us which areas are active. All right, so with that, I'm going to move on to the next slide. Let's start taking those elements that we looked at and start piecing them together. Let's start putting them together in molecules. Those molecules that we'll be looking at, now we've taken 2 elements together. We want them to hold together. And the 4 types of holds that you'll see on here are chemical bonds. And they'll rigidly hold, well, maybe not all rigidly, but at least hold the molecule together. The 2 of which we'll talk about take place within the molecule. They're called ionic bonds and covalent bonds. The third one are called hydrogen bonds, which attract each other. And again, I'll talk about these attractive forces in a little bit. And then metallic bonds, not so physiologically relevant, so I won't really talk about these. Okay. So you guys can refer to this slide because I'm going to talk about these in the next couple slides. So ionic bonds, complete transfer of electrons. Let's take a look at that in sodium and chlorine. You guys already did this for me. Going back to this prior slide, we looked at sodium and we looked at chlorine, of which we know sodium has 1 electron on the outer shell, which you guys already told me. I told you guys there's 7 electrons on the outer shell for chlorine. So let's take that example where we mentioned earlier. The thing is about atoms is that they are most stable when they have a full shell on the outer shell. So looking at sodium, you see, as I said earlier, first shell, 2 electrons. Second shell, 8 electrons. Outer shell, there's 1 electron. So in order for it to be stable, it would most likely get rid of that excess electron. So if each electron has a negative charge to it, and that sodium gives off an extra electron, is it going to have a positive charge or a negative charge? So it's going to have a positive charge. So that positive charge on here is called any charged element is going to be called an ion. And ions can be categorized. Again, not something you really need to know, but we have cation and anion. Cations are for positive ions, anions for negative elements. And a way to remember this, if you were to think, if you're a cat person, cat would be positive. Anion kind of looks like onion. Onions are negative. Unless you really like onions, then you got to think of something else. But those are the names of those types of ions. So in this case, that sodium ion would be a cation, and it has a positive 1 charge. It gave off 1 electron, so it has a positive 1 charge. So we typically would see Na+, and that plus signifies 1 electric charge, unless it's something like calcium, which has 2 electrons that would give off. Again, I digress. Now, if we look at chlorine, chlorine just needs 1. It has 7 on the outer shell, so it needs 1. So its likelihood is that it would pick up an electron. So if it picks up an electron, would it be negative or positive? Right? So it's picking up an extra electron to fill that outer shell. If it picks up an extra electron, it would be negative. In this case, it would be called an anion. So chlorine, Cl, would have a negative to it. So given that, we know opposites attract, right? So positive and negative ions happen to bond with each other. And that's exactly what's happening in this case. It's called an ionic bond, where you get sodium that gives off an extra electron. And now it's positive. Chlorine picked up an electron. It doesn't have to be from that original sodium. It just picks it up from anywhere else. Picks up an electron, becomes a negative, an anion, and that cation and that anion bond together. And that's an ionic bond. And if you were to take a look at a bunch of sodium and chlorine atoms, they just happen to kind of attract each other and kind of cluster together. So this would be sodium chloride, which you might already know is table salt. It's exactly what it is. So that's ionic bond. On the other hand, we have something called covalent bonds. While ionic bonds resulted in the transfer of electrons somewhere where you had a positive charge and a negative charge, covalent bonds actually share the electrons. Okay, this is a different type of bond. And to give you an example, let's take a look at hydrogen atoms. Hydrogen atoms have on the outer shell only one electron. And as I mentioned earlier, the first shell needs 2 electrons. So in this case, that hydrogen atom, this hydrogen atom comes together, this hydrogen atom comes together, and what they end up doing is they share one shell. So at one point, the electrons over here, 2 electrons on this side, and then those electrons zip to the other side, and it satisfies both. So you get these electrons that are literally racing around their racetrack, being able to fulfill. So by sharing, they can fulfill each other, and therefore these bonds are formed just through that sharing of those electrons. In these cases, you'll see the term nonpolar, and I'll talk about that in just a little bit. Let me give you the example of oxygen. Oxygen is stable if it can share, if they can each share a pair of electrons. So here you'll see 4 electrons, it needs 2 on each side, so it ends up sharing 2 pairs of electrons. So you'll see those electrons forming back and forth, and what you see on here, so now that these oxygen atoms are now satisfied with the number of electrons on their outer shell, this forms a double bond. So this is a double covalent bond. Something we'll see with oxygen later on. Now let's bring it back to carbon. Taking a look at carbon, you'll see 1, 2, 3, 4 on the outer shell. So the fact that there's 4 on the outer shell, it would need how many electrons to satisfy the outer shell? It needs 4, right? So if it needs 4, it means it can form 4 bonds. And in most cases, that carbon will form a bond with hydrogen atoms. Those hydrogen atoms only need 1. What they can do is sort of share one of those bonds with that carbon. Another hydrogen can come and share, and 4 hydrogen atoms can come share together. So at this point right here, you can see our carbon atom. Carbon is now sharing electrons with 4 hydrogen atoms, and this becomes our methane molecule. Methane such as methane gas. So carbon I'll talk about a little bit because carbon is an interesting atom for having 4. It allows us to build these huge molecules. Again, I don't want to spoil the fun on that, so I'll come to that in just a little bit. So I mentioned nonpolar because if we take a look at that racetrack that we just built around that hydrogen atom, those electrons spend equal time on both sides of the hydrogen atoms. They'll literally just race around the racetrack, and there's no point at which the electron spends more time around the hydrogen or the other hydrogen atom. That'll probably be better explained when we take a look at the next example of a polar covalent bond. And the best example for this is oxygen with 2 hydrogen atoms. So oxygen needs 2 electrons. Hydrogen has an electron that it can share. So in this case, oxygen binds to 2 hydrogen atoms. However, oxygen is a big atom. Same with phosphorus. And remember this because we'll be able to see this a little bit later on. Oxygen and phosphorus have a tendency to be electron hungry. They have a tendency to kind of pull electrons towards them. And if you look at the hydrogen atoms that form together, we create H two molecules of hydrogen with one oxygen atom, H2O, otherwise known as a water molecule. And the sharing, the covalent bond that's sharing, we still see that those hydrogen atoms still share their electrons with that oxygen atom. But if you were to draw this racetrack, and you were to draw that electron that's moving around, which part do you think it would spend more time around? The hydrogen or the oxygen? Well, let me even draw something where it would really look. Draw that racetrack. Now, where do you think it's spending more time around? Oxygen. It's spending more time around the oxygen. So if I have two electrons and they spend more time around the oxygen atom, that has a tendency to make the oxygen a little bit more electrically negative. So if you spend more time around one side, it's going to make that electron spend more time around here and become more of a negative charge. And that term we call electronegativity. And that has a profound importance in the structure of the water molecule. In this case, the oxygen atom would be electronegative, which you can see with the signal over here, and that's a sigma signal. Electronegative. And conversely, because the electrons spend less time around the hydrogen, they have a tendency to be electropositive, which you can see again with the symbol above. So, because the electrons spend more time around the oxygen, that produces a polar covalent bond, or a molecule with electronegativity, a side that happens to be a little bit more negative. And that, when you look at other water molecules surrounding, this is electronegative, and this is electropositive. If I were to take another water molecule, and this happens to be electronegative, and this is electropositive, again, opposites attract. Electronegative and electropositive creates a little bond. And that bond is hydrogen bonding. These are weak bonds that form between the electropositive hydrogen atom with an adjacent oxygen atom from an adjacent water molecule. And these are weaker bonds than the covalent bonds and the ionic bonds. In fact, they're fairly weak. But each oxygen atom can form 4 hydrogen bonds, 1, 2, 3, 4. So if you think of water molecules and they're constantly kind of pulling on each other, there's a lot of properties to water that you can see. And you can see that on our next slide. The first 2 properties I want to talk about. I want to talk about cohesion and I want to talk about adhesion. And cohesion is water sticking together. You think of a couple and they might be cohesive. It's because they stick together well. On the other hand, you have adhesion. Adhesion is like an adhesive, like a tape that sticks to something. So in that case, tape would stick to something. In this case, what I'm talking about is water sticking to stuff. So water has that property of this stickiness, and that's related to the hydrogen bonding. So you'll see that since water sticks together, here you can see a water strider. This is a specific bug that walks on top of water. And the reason, if you take a look at the surface of the water, you can see that it kind of bends in a little bit. Those water molecules are holding together. Those hydrogen bonds happen to hold the water together, and the water strider can run across without breaking the surface of water. It's that light. It can do that. If you take a look at little dew droplets that form in the morning, that's the water vapor in the air that just happens to collect on surfaces. That's adhesion. That's water sticking to stuff. And then all of a sudden, the water droplets surrounding stick to one another and they start to become cohesively stuck together. Another example I want to use, if you ever wonder, I don't know if you guys ever wondered this, think of a plant and you water the base of the plant. have you ever wondered, how does water get up the plant? How does it counter gravity to make it into the plant? Would you think you would probably want to pour it on the top to see if it somehow simmer down? Well, no, that's not the case. If you were to take a look at a stem of a plant, and here we have the roots, and here we have the water, and then right on top of the plant we'll draw one little single leaf. We have a bunch of water droplets that will start to stick to the inside of the stem. And what happens when you get sunlight that hits the top of the leaf, we know that's going to cause evaporation of water. Water starts to fly off. And as water flies off, what that ends up doing is it ends up pulling a whole string of water molecules up due to those cohesive properties. And it will pull from the roots and counter gravity. So that's a cohesion of water molecules pulling up that whole string of water beads all the way up. On top of that, if you remember from, if you ever measured things in a graduated cylinder, you probably ever took a look and you're trying to measure the amount of fluid, you saw that the edges kind of curve up against each other. That was a property of adhesion where water molecules stick to the surface of that cylinder. And here, if you remember, where were you supposed to measure? Well, it's due to the meniscus. That was what we called the meniscus. It was this curved area. That means that water happens to stick to the sides. So that plant happens to climb along. If you ever take a look at the straw, it happens to do the same thing. It literally will pull on the side, attracted to the stem walls, and attracted to one another as that water gets pulled out. So that's adhesion and cohesion. Other things about water. It is a universal solvent, meaning you take anything like salt, you take anything like sugar, you take anything like glucose, you take anything and you put it in water, and over time it will dissolve. It works really well for that. In fact, if you were to take a look at water, and I were to take NaCl, which was our salt, and we take water starts to come in and surround, ends up surrounding that chlorine ion because of the electropositive and the negative. So the electropositive negative have a tendency, and you'll see that hydrogen the water molecules surround that chlorine ion. So it'll break it apart. Water also has high thermostability. What do I mean by that? Water has a tendency to hold heat very well. If you ever think about why you sweat because you're generating heat. And the best way to get rid of that heat is a process of sweating. So that when you start to sweat on the surface of that skin, as water starts to pick up that energy that's being provided by that heat that you're generating, those water molecules start to jiggle around. And they start to break these hydrogen bonds, therefore releasing a good amount of heat. And as that water molecule comes off, you remove a certain amount of heat. Now that we're kind of like a little bit in a heat wave, do you guys remember ever going to like Magic Mountain or any like theme park and you step under those little mister things? Why does that cool you if they're just sprinkling just normal water on you? Well, when you stand underneath it, that water sticks to your skin, and eventually you heat it up and you evaporate. If you ever look at swamp coolers are some of, I don't know if you still see them around anymore, but basically it's a big fan and it blows mist on you, and it should eventually just cool you off. Another thing to look at, if your engine ever overheats and you need radiator fluid, what's something you can use? You can use water. Water works so well to be able to hold heat. In fact, let's even take a look at temperatures. Has anybody been to the tropics anywhere? Like anywhere with high humidity? Well, how's the temperature over there? Always hot, right? At night, it happens to also be hot. It doesn't really cool down. And that's because the amount of water vapor in the air because of the high humidity, there's a bunch of water vapor in the air, and that water holds that heat well. So that when it turns night, the temperatures don't really fluctuate much. If you haven't been to the tropics, you've probably been to Vegas, right? You have less people there. We'll get more people that admit they've been to Vegas. In Vegas, you notice, especially in summer, it can be 110, 120 during the day. but at night it drops down to like 80, 70. Right? So there's a big discrepancy between night and day temperatures. And that's because of the less water vapor in the air. So desert temperatures have a tendency to fluctuate quite a bit. Tropics, due to that humidity, have a tendency to stay. So that's the thermostability. We'll talk a little bit about reactivity and how it interacts with reactions taking place. And water also serves as a great lubricant. All right, so now that you have that, we've talked about the molecules. We've talked a little bit about building molecules. Let's start talking about chemical reactions, where we start taking these molecules and have them start reacting with each other, with the whole, and tying in the concept of what happens to that energy. Because this is going to have a profound influence on how when we eat, we take energy from it to use it to couple with the energy that we need to survive. So first, before I want to start out, I want to kind of talk about the key players involved. We have reactants and we have products. And any chemical reaction, I'm going to give you an easy example. A plus B forms C. And you'll see that the reactants are the starting substances and products are the ending substances. So if I see A plus B, are these my reactants or are they my products? These are my reactants. And I know C will be my product. It's the ending substances. And in these reactions, there exists energy in the system that we can quantify. The first one we'll measure is potential energy. And potential energy is energy that's stored, and I use this term loosely, stored in the bonds, whether they're ionic bonds or covalent bonds. The real term for it, the potential energy, is the amount of energy stored as that state of being. Kinetic energy, on the other hand, is the movement of matter. So that's molecules moving around. They happen to kind of just jiggle around in their space. And the more they jiggle around, the more kinetic energy it has. So looking at that, if I were to take a look at, for the example of a roller coaster, and at the very top I have that roller coaster cart that's just sitting right at the very top. It's not moving at all. It has a high potential energy, in that that's the energy that it could use to start to fall down. So if I were to take my remote here, it has a bunch of potential energy. It's not moving, so its kinetic energy is 0. But if I let it go, and it's about to hit the ground, that potential energy is decreasing, while the kinetic energy, the energy of movement, is increasing. So in that case, you'll see what I was talking about earlier. High potential energy, high potential energy, low kinetic energy. And when I drop it, when that roller coaster gets down to the very bottom, let's just say low potential energy, high kinetic energy. The energy transfer that happens back and forth is conserved. And that is our first law of thermodynamics. Whenever we look at a system, when we move from the reactants to the products, the energy is always conserved between the two. There's no loss of energy, and there's no gain of energy through this reaction. It's always going to be transformed from one to another. But if you're to use that example and you think about that roller coaster, if that were absolutely true and that were the only law of thermodynamics, that would mean that this thing could go on forever and it would non-stop move over and over again. But there's a little bit of loss to this energy. And I want to use, again, that term loosely, of loss of energy. What's given off over time? If you were to touch the wheels of that roller coaster cart, it would be hot. There's a certain amount of heat that's given off. And that's part of the energy transfer that occurs. There's a loss in potential energy that happens due to heat loss. If you ever drive your car for a long distance, you know your car gives off a certain amount of heat. But if you were to measure that energy that moves from this point to this point, and also add the amount of heat loss, that energy from this point to this point. Potential energy, kinetic energy, plus the heat that's given off. The potential energy in this case, plus the kinetic energy, plus the heat, those two energies, the sum of both energies, would be the same. So that's the second law of thermodynamics. Another idea is to take this again into something physiology related. If you were to think about the food you just ate, right? However long ago, you take the energy from the bonds that are in that food and you break it apart, and then you use that energy. You don't actually use all of that energy that you take from food. Some of that is actually given off as heat. If you don't believe me, feel your own skin. You're giving off heat. That heat, in fact, we're not even really all that efficient in terms of converting all of that food to potential energy that we can use for growth, reproduction, movement, building of proteins, building of stuff, repairing ourselves, etc. We actually give off a good amount of heat. In fact, if the amount of food that you eat, 25% actually gets stored as potential energy. The other 75% we actually give off as heat. And we do that for two purposes. Number one, to keep your, well actually I only know one right now, is just to keep your body temperature at about 98.6 degrees. So that's the second law of thermodynamics, is the decrease in potential energy. If you go into the book, it talks about entropy, and that's entropy is a measurement of the measure of disorder. I don't want you guys to get too lost on that concept, but basically when heat disperses, it will travel. Like if I give off a certain amount of heat right here, or if I were to have like a cup of boiling water that's giving off heat, eventually that heat will continually disperse. That's the disorder that comes from it, and it will disperse into this room until it's all equal. The third law isn't even really physiologically relevant, but it's basically us saying we can't get to the coldest temperature ever, which is like absolute zero. That comes to a certain point. It's a theoretical point where these molecules have zero kinetic energy. And the molecules, they normally jiggle around, and eventually you get cold enough that the molecule just stays still. And that we can't really get to. We've had laboratory settings where we try and get as cold as possible, and it's really, really close. We just can't get to that point. It's almost like, I guess in ways, kind of like stopping time. But again, I don't want to digress too much. Okay, so now I've talked about this as a recap. A plus B are reactants, and C is our product. And I want to use another example to help really tie this in. And that's going to help us explain some of the other concepts that we're going to be looking at. Because what we're doing is we're going to be building on top of this. A plus B, we're just going to use the example, and I'm actually going to just draw this on the board. We'll do A plus B, where A equals man and B equals woman. And then these are our reactants. And then C equals baby. Okay? And this will be our product. So again, I'm just using this as an example. But in this case, A plus B becomes our product. So in this example, you just have to pretend that man plus woman form together and they produce baby, and then the man and woman are expended in them. They just disappear into the thin air. They get destroyed in the process. So man plus woman becomes baby. So think about that as we move on to the next slide. So in this case, we're going to take a look at the amount of potential energy in a reaction. What's needed in order to go from man plus woman, which are reactants, to turn into our final product called baby. And there's a certain amount of energy that's going to take place. There's an energy transformation, where if we measure the amount of energy here, plus the energy here, they're going to be equal to one another. So in order for this reaction to take place, we have to measure the amount of potential energy. Just like we saw on the prior slide, in order to get from here to here, we want to measure the potential energy at section A, and we want to measure the potential in section C. That gives us the sort of progress on this reaction taking place. So what I want to look at is the measurement of potential energy for A plus B and then our final energy in our products with C. So man plus baby, here's the energy of the reactants, and then C is the energy of the products. And you're probably wondering, well, didn't you just say that the energy should be equal? Well, that's true. But there's a loss in potential energy, which means there must be a gain of some form of kinetic energy. And that energy could be heat in this case. And so this, from here to here, this is the level of the products, and this is the energy level of the reactants. This right here is a negative value. This is the amount of energy that's lost. And you don't really have to know this term, delta G, but that's the change in potential energy. And we have a negative change in potential energy. So by measuring the potential energy, we would see that typically in this case, and let's use that example earlier at some point. A plus B, you have a bunch of A's and B's in a cup. Let's just put a bunch of men and women in a beaker, or a room, or whatever you want to call it. A bunch of A plus B, A, B, A, B. And think of these men and women just going around, randomly just moving around like this. They're just floating along, and eventually, they might just get in the right position and bump into each other and start a reaction. And that reaction, when A and B come together, they form what again? They form C, they form baby. So now we're going to wipe that out, and we're going to form now C. Okay. So the amount of energy that's required, there's a certain amount of energy that you need for those two to get together. They don't just bump into each other and just form baby all of a sudden. They have to get into the right configuration, the right place, and eventually there's a certain amount of energy that's required to start that reaction. If that doesn't make sense, think of a fire. I have a bunch of paper on my desk right now, and I want it to catch on fire. It's not going to just spontaneously catch on fire. There may be an off chance that somehow some ray of light comes in and might spark something up, but really what's needed is some form of energy to get the reaction to start. It's just a little bit of energy that you need to invest to get it started. So in this case, I might have to use a match just to get it to light up on fire. Or if I were to take that roller coaster, for instance, that roller coaster I mentioned earlier, that thing might just be sitting there. And in order for it to get down to this point, you gotta wait some time. Eventually, maybe if the conditions are right, maybe somehow it just starts and then it starts to fall. The activation energy is that little push that's required just to get that energy started. That means you need to put in a little bit of energy in order for those things to occur. So in this case, the reactants in this case have to have a little bit of energy to get into the mood to eventually be able to form our product. Now think about things that would speed up the process. And what I mean by speeding up the process is the speed at which this reaction takes place. A plus B turns to C. If I start increasing the number of our reactants, putting a bunch of men and women into this beaker, there is a higher likelihood that our products start to form at a faster rate. Why? Because there's a bunch of them. There's a likelihood that they'll just bump into the right configuration, and then these form a reactant, or a product. And then these form a product. And then these form a product. So that speeds up the reaction. You want something to happen a little bit faster, add more reactants to it. Something's not burning fast enough, the paper's not burning fast enough, add more paper to it. That's one way. The other way is to increase the temperature. If I increase the temperature, start moving around faster, and they move around faster, and they move around faster, and eventually there's going to be enough energy that they bump into each other fast enough that they create products faster. If that concept doesn't make sense, think about what happens when you're trying to dissolve a bunch of sugar in water. You think this is taking a long time. What happens if you heat that water up? You start introducing heat, those water molecules start moving around, and then whatever your sugar is starts to get dissolved faster. Because those molecules bounce around faster. So increases in concentration and then increases in temperature will speed the reaction further. And then to even take it a little bit further, let's say if I were trying to slow down the reaction, and I've got a bunch of, it's been some amount of time, and eventually I've got a bunch of product around. Do you think this is going to speed up the reaction or slow the reaction down? Let's slow it down. Because A can't bond with C, B can't bond with C. These are our products. It's only man plus woman that will start to form baby. We can't do man plus baby. That doesn't do it. So, in this case, the rate of product formation, when we start to have a bunch of products in there, that slows the reaction down. That, if you go into the book, is Le Châteauneuf, and I can't pronounce that right, or even spell it right, but you get the general gist of it. The principle in that A plus B forms C, you increase the amount of C, and that slows the reaction down. You get less A and B. You want to speed up the reaction, get rid of the babies in there, and eventually you'll get more reactants that bump into each other. What questions do we have about this concept before I continue? No questions? All right. So remember this slide, because I'm going to pull up this in just a little bit. We're going to add on to this concept. So now that we've talked about energy transfer, let's look into ways energy can be transferred in or out of our reactions. And if you're to take a look at the two types of reactions we have endergonic reactions and we have exergonic reactions. And the main difference between the two is the direction of which energy is going into the reaction or exiting the reaction. Endergonic reactions require energy to be put into the reaction in order for that reaction to take place. Exergonic reactions are reactions in which one of the products is the release of energy. So looking at that example I talked about earlier, A plus B plus a certain amount of energy produces baby. This is baby turning into man plus woman and releasing a good amount of energy. To kind of give you an idea, this is how fuel works. This is how dynamite works. There's a good amount of energy in there, and once you start to break them apart, you release a good amount of energy. And that energy that's being released is released from the bonds that are formed within the baby, typically where that stuff happens. To give you another example, this is that hamburger. Now I'm just thinking hamburgers, I had In-N-Out last night. That hamburger being broken down and releasing amount of energy from it. I'm breaking that hamburger down into its smaller constituents and releasing energy. This endergonic reactions is where you have energy being put into the system. So before I go a little bit further, let me help you clarify these two terms, endergonic and exergonic. How can I remember the difference between endergonic reactions and exergonic reactions? I highlighted ender and exer for a reason. Ender for entering energy. Exer for exiting energy in the reaction. That's another mnemonic that I'm giving you. I think this is number 3 now. And in endergonic reactions, you'll see these are typically referred to as anabolic reactions. We call it anabolic, that's the term anabolic, like anabolic steroids to build. It's exactly what we're doing. We're taking small components, A plus B, and forming it into one bigger molecule. So this is an anabolic reaction. And in this case, in order to get an anabolic reaction to take place, you got to invest some energy into it. It's an endergonic reaction. And in the same token, from looking at from an energy perspective, you also see the term dehydration synthesis. Dehydration synthesis. This is the synthesis of molecules that requires dehydration. So in this process, do you think you're losing a water molecule or you're gaining a water molecule? You're losing a water molecule. So this is loss of water. Again, not wholly important, but I will show you in some of the reactions, I'll show you where there's a loss of water molecule to put these two together. Now, exergonic reactions are catabolic reactions. This is where you're breaking apart those bonds. Yes?
Can you, where this is synthesize a large molecule from a simpler, smaller molecule? Like, I don't understand that. Like, making simpler together.
Putting a water molecule together, or putting molecules together.
So basically, you're starting from creating a bigger molecule by putting smaller molecules together.
Yeah, like think of like muscle, for instance, putting proteins together. Think of building a starch molecule. You're putting things together. You're constructing them. You're taking, and we'll be able to see these molecules a little bit later on. So hopefully that'll clarify that in just a minute. Now, the exergonic reactions is the release of energy. So in this case, we call them catabolic. So this is breaking them apart. And in this process of breaking apart those bonds in a molecule, you're releasing energy. And that release of energy can be used to be coupled with another type of reaction. What do you think that would be? Well, if this is releasing energy, this might be requiring energy. So that same energy where you're breaking apart energy allows you to create endergonic reactions that require energy. So think of what you're doing when you're trying, let's say you have cells that are growing, replicating. They require energy. So the breaking apart of energy, if you're going to take sugar molecules or molecules from the food that you just ate, you're breaking that apart to couple that with reactions that require building. So the combination of endergonic and exergonic reactions are exactly what metabolism is. And we're going to learn more about this in next Thursday's lecture when we talk about cellular respiration, which is the process of breaking apart a glucose molecule. We're breaking apart a glucose molecule into its smaller products to release a good amount of energy. And that energy, which we'll talk about next Thursday, is a molecule which we'll call ATP. Again, we'll talk about in a little bit in today's lecture. It's a form of energy. So in this case, you'll see this is in exergonic reactions because it releases energy. It's called hydrolysis. The term lyse, again, something I'd like you guys to be familiar with. It's something you're going to be able to see later on. Lysing is to break something. And it requires hydro. What do I mean by that? Water. Water lyse. Water molecule is introduced into a molecule along to start the reaction to break it apart into its smaller constituents and release energy. So now that we've talked about the reactions, the reactions are where we have endergonic and exergonic reactions. Let's take a look at molecules. We'll take a little jump and take a look at the molecules that can be formed. And again, we can break these down into inorganic compounds and organic compounds. Inorganic compounds are molecules that lack a carbon backbone. This will make much more sense when we look at our carbon-based organic molecules in just a bit. But inorganic compounds lack that carbon backbone. It doesn't mean they don't have carbon, it just means it doesn't have this carbon structure. So with that, we can see what types of molecules we have. We have water, H2O. We have acids, bases, and salts. And vitamins and minerals typically are typically inorganic. So here you can see with acid, bases, and salts, something I won't really get too much into, because again, this is something that you saw from chemistry. We can measure acids and bases. Acids in a process that releases hydrogen ions. This is an example of an acid. Base, which contains a hydroxide anion that has a negative charge. And we can measure the pH of a sample by measuring the concentration of the hydrogen ions. Think of sulfuric acid, you think of carbonic acid, you think of just battery acid, you think of any type of acids, and they give off these hydrogen ions. And we can measure that. And in fact, pH is on a negative scale, meaning the lower or the higher the acidity, the lower the pH. It actually exhibits a logarithmic scale, which you don't really need to know, but pH, neutral pH would be a pH of 7. That means this is fairly neutral. We know that things that have a higher pH are more basic. So anything about 8 plus is going to be basic. And anything below 7, anything less than 7, is acidic. So we can measure that based on that pH. And the importance behind this, the importance in our physiology, is just like we talked about on Tuesday. We have a narrow window that we typically will consider normal. And blood has a pH of 7.35 to 7.45. If you don't believe me, take a look at your labs when they're drawn, and they will measure the pH of your blood. Anything below 7.35, remember, negative logarithmic scale, anything below 7.35 is acidosis. Anything above is referred to alkalosis. So, low pH, acidosis. Higher pH is alkalosis. And if you were to measure somebody's pH of their blood and it drops, that's a condition called acidosis. And they typically want to find out what's contributing a bunch of hydrogen ions into the bloodstream that's causing it to be slightly acidic. And there's metabolic acidosis, there's respiratory alkalosis, there's a bunch of causes for the disruption in the pH. And it causes a whole host of changes that happen physiologically, which we won't really get into right now. But we do have a buffer in our blood that prevents the changes. You would think, well, that means if I just add a little bit of acid to my blood, is that going to change my pH? Does that mean I'm going to die? No. We have buffers in our blood that prevent minute changes in the pH. And that buffer is bicarbonate. This example right here, H plus HCO3 becomes carbonic acid, is something if you don't get now, that's okay. Because we're going to beat this whole concept down over and over and over again. This is the fundamental concept in physiology. It's carbonic acid. When you get CO2 plus water, it forms a weak acid known as carbonic acid. And that carbonic acid in water can dissociate into two ions, a hydrogen ion and a bicarbonate ion. That bicarbonate ion can attach to other hydrogen ions. So if you were to inject a little bit of acid into somebody's blood, those bicarbonate ions would attach to it to neutralize that acidity. And then, what happens with that other hydrogen ions? Well, we can get rid of this in multiple ways. You'll learn in our respiratory lecture, we can get rid of hydrogen ions this way. You'll learn in our urinary lecture, we can get rid of hydrogen ions through urine. You'll learn through the digestive system, we can get rid of hydrogen ions there as well. We have multiple ways of getting rid of it. That's the whole focus of what we're going to be talking about in our urinary lecture, is the getting rid of acid. But what's left over is this bicarbonate ion. That's that buffer that prevents any changes to keep us within that narrow window of 7.35 to 7.45. So that is our example, our first example of homeostasis in blood. And again, we'll go over this in our cardiovascular lecture when we talk about blood. Okay, now on to organic compounds. Organic compounds are compounds that contain a carbon backbone. And what do I mean by carbon backbone? Remember what we talked about earlier about carbon, where it contains 4 electrons on that second outer shell. It can bind to 4 other bonds, 4 covalent bonds with another atom. And in most cases, you'll see that it forms with carbon molecules. It's our way of, if you were to take an example of aliens, if aliens existed and they came down and they sampled humans and they found out what causes, not even just humans, but all life form on this earth. We know that the earth is comprised of 70% water. But what makes us really unique is that we are carbon-based forms. We have carbon atoms that attach to one another to create these huge molecules. Okay? It's one thing when they're looking at Mars and they're trying to find water, and they say, okay, water, great. Water is something because it has those properties that we just talked about. But then the next question, are there organic compounds that exist? Because that means if there are huge carbon molecules, that means there had to be some chemical reactions that took place to create those carbon atoms, to create these huge organic compounds. And if something had to create it, that means that something could be sped up with stuff that's produced, possibly by life. Again, I don't want to digress too much. I want to focus specifically on our organic compounds. But carbon can bind, let's say, to another carbon atom, and that forms a covalent bond, C and C. And that's one covalent bond that forms. And now, one thing I want to point out is now we have 3 electrons from each carbon that can bind together. Typically, what would happen is hydrogen can bond to each of those free bonds, or free electrons. And I'll talk about that in our next slide. That's our alkanes. You'll see that the formation of these huge carbon atoms, and in fact, that C can bind to C, can bind to C, can bind to C, can bind to C, and you get a bunch of H's wrapped around. You guys get the idea. Another way of drawing it, if you remember from chemistry, is that you can draw these signs. You guys remember these? And each of these points right here are carbon atoms. So this serves as the foundation for huge molecules that we can create. And the 4 main organic compounds that we'll be looking at are carbohydrates, proteins, lipids, and DNA. So this is kind of funny, because whenever you go to the grocery store and you see the term organic, typically if you were to see organic, you kind of want a definition of what they mean by organic, because this is a term you've probably seen abused quite a bit. Just because a fruit is organic, yeah, of course it's organic. It contains organic compounds in it. There's a bunch of sugar molecules in it. That makes it organic. But that's not the term they might be referring to. Organic referring to as no pesticides being used or whatever other organic things are being terms are being used. That's the whole concept behind organic. It's just carbon backbone molecules contained within. So with that, now I want to take a look at that molecule that I pointed out earlier, our carbon that's bound to a bunch of hydrogens. This is what we call alkanes. And this is what's called hydrocarbon. They're called hydrocarbons because we have a bunch of hydrogen and we have a bunch of carbons. And it's exactly that molecule that I just drew over here. I have 2 carbon atoms bound together by 1 covalent bond, bound to a bunch of hydrogens surrounding. And in this case, if you were to take a look at 2 carbons and 6 hydrogens, 2 carbons, 6 hydrogens, you have what's called ethane. If you get 3 carbons and 8 hydrogens, you have propane. You have methane, butane, pentane, hexane, octane. These are all your fuels that you would have heard of. These things burn off and release a good amount of energy. You've probably heard of octane. This is stuff in gasoline when they're measuring. That's our hydrocarbon. Now we're going to take that same structure that I talked about earlier, and instead of hydrogens on one of the connection points, I'm going to leave this one free. There's a weird thing on my computer that it just keeps erasing. Anyway, there's this point right here that we're going to just talk about right here. And I mentioned this on this last slide right here. Functional groups. Functional groups is a way of taking a molecule, taking one of those slots that attaches to carbon, and attaching a group on there that would change the characteristic of that molecule. of which we have 1, 2, 3, 4, 5 main ones that we're going to be talking about. These are things that can attach to that point right there. And one thing I'm going to talk about is one of the functional groups is OH, a hydroxyl group. So instead of a hydrogen, I'm going to put OH on there. And in that functional group, we have OH. That becomes our alcohols. So if I have ethane and I attach a hydroxyl group, which makes it an alcohol, this would be called ethanol. Has anybody heard of ethanol before? Ethyl alcohol. That's the stuff in alcohol. Right? That's ethanol. And that changes the characteristic. Where this was a pure fuel, this becomes an alcohol, which also serves as a fuel too. The other thing that we have carbonyl groups, and you'll see some carbons that have a double bond to an oxygen. Another one is a carboxyl group. And that is a carbon double bound to an oxygen bound to a hydroxyl group. So these are typically called acids, a carboxylic acid. Typically they would be acids because that hydrogen can break off and create a hydrogen ion. But another way of remembering carboxyl group is to, let me just erase this H real quick, draw it up here, and this creates a little box, carboxyl group. Amino group is another functional thing that we're going to be able to see. Take off that hydroxyl group and add an amino group. And this is going to serve as a foundation of what we're going to be talking about later on. So just remember this molecule for now when we talk about this towards the end of our lecture. This is going to serve as the foundation of protein. And this creates an amino acid. The last group is sulfhydryl, SH. And sulfhydryl groups contain sulfur. So typically compounds that contain sulfur on it have that smell. Do you guys remember those, I don't know if you guys ever played with these, they were like the stink bombs? Yeah, and you drop them, and then you get this concentration of sulfhydryl molecules in there that start to disperse, and once you pick up on it, you smell the smell of rotting eggs. Sulfur is that exact smell, and that's a way you can remember a sulfhydryl group is SH. If you just write IT at the end, you'll remember what it smells like. That's exactly it. Okay? When we get into our, specifically our sensory lecture, we'll take a look at the molecular compounds that are found in things that smell. Body odor. You'll see tons of sulfhydryl groups on there. But sulfur doesn't just form the process of smelling like things. In fact, something that you will see in especially molecular biology is that it forms very strong bonds called disulfide bridges. Something that you will really see, especially in our protein section that we'll talk about later on. You get one sulfhydryl group and another sulfhydryl group, they can break those apart and create these strong bridges that will hold a molecule together. We were able to take a new functional molecule from that addition of that functional group. We now have a new molecule. So what we're going to do is we're going to talk about 4 main macromolecules. And if you remember which ones they are, we had carbohydrates, we had lipids, we had proteins, and we had our DNA, RNA. These were our and carbohydrates are going to be the first one I'm talking about, of which you're going to be able to see on here. An example of one of our carbohydrates. If you're thinking carbohydrates, yes, these are the same carbohydrates that you see on the labels of food. This is our way of measuring, or our way of knowing that this is a specific carbohydrate. And the way we can know on that structure, typically what you would see are hydroxyl groups attached to a carbon backbone. You would typically see those, and something that you would also see that's characteristic about carbohydrates is that they can form rings. So you get sugars that typically have 6 carbons on them. Before I do that, I want to get into the concept of a monomer and a polymer. So the term we're looking for on here, the prefix is mono, referring to one. And polymer, referring to many, to the poly. And so when we look at monomers and polymers, monomer is a single unit. A polymer is a unit with a bunch of those single monomers attached. And when we look at carbohydrates, we have polymers that we'll be talking about. But first we have to start off with our monomer for carbohydrates. And these are what are referred to as simple sugars. Simple sugars because it contains one saccharide. And the suffix here, saccharide, which you've probably heard the term for saccharin, which is sweet, means one sugar, hence the term simple sugars. And these simple sugars, you can see we have 3 types of simple sugars. We have glucose, we have fructose, and we have galactose. Glucose, of which you may have heard of, is something that we use as a readily formed energy source. Fructose is a simple sugar that's found in fruits. Have you ever heard of high fructose corn syrup? That's a bunch of fructose all crammed together. Fructose, if you were to taste glucose, has a sweetness to it. If you tasted fructose, fructose in the same weight is much, much more sweeter. Hence why if you were to look at why we use high fructose corn syrup as a sugar substitute, it's because you can get a lot more bang for your buck. When you can create a bunch of fructose, it tastes as sweet, you can use less of it, but still not good for you. Galactose is the other component in milk sugar that we won't really talk too much about, but it's that third monosaccharide. Now disaccharides, on the other hand, are two sugars attached together by a glycosidic bond. You don't need to know the term for that. Glycosidic bond is just a bond that forms those 2 sugars together to create a disaccharide, of which we'll talk about 3. Number 1 is sucrose, which is a combination of glucose and fructose. That forms table sugar. Anytime you buy sugar, that's what it is, glucose and fructose combined together. If you look at lactose, lactose is a milk sugar. That if you read milk and you look at the back label and you think, oh, this is high in sugar. What exactly are those sugars? That's lactose. And lactose is a combination of glucose and galactose. Is anybody here lactose intolerant? Really, that many people? Just 4 of us? Okay, well, lactose intolerance is the inability to break down glucose or break down lactose into glucose and galactose. And we'll talk about why we have that inability. Maltose is grain sugar. If you hear of malt, like malt liquor or malt ice cream, that's basically the sugars that come off barley. You'll see a lot typically in barley or other grains, you extract the sugars. If you ever think of malt liquor, what they're taking with those maltose is they sit there and they ferment it over time and it turns into some form of ethyl alcohol. And the reason I'm kind of going to more examples of this is because I took a beer brewing class not too long ago to kind of burn 4 hours of time. But learned a lot about it, but never relevant because I actually never made anything. But nonetheless, that's maltose. Glucose plus glucose. Polysaccharides is a string of sugars all put together. And if you think about a whole string of sugars, think of like a Pez container. You have a bunch of glucose molecules strung together. Those are your polysaccharides. It's a polymer of a bunch of carbohydrates. And this is what's referred to as complex carbohydrates. So if you ever look at, again, the back of a food label and you wonder what's the difference between simple sugars and complex sugars or complex carbohydrates, that's exactly what it is. It's a bunch of carbohydrates strung together. And how we store our energy, if you think of why do we need complex carbohydrates, it's how we store energy. We store it in the form of, and I'll talk about this in just a little bit, it's our storage form. And in fact, plants do the same thing. They store a bunch of glucose molecules together. So the first one, let me first refer to as starch. Starch, we know high starchy foods like potatoes and can't think of any other starchy foods, but other high starchy foods. They contain a bunch of complex carbohydrates. And why? Because they string a bunch of glucose together in starch molecule. So that way, and you'll learn about this in cellular respiration, when that plant needs to burn a glucose molecule, it does it by using that starch. It breaks down that starch, that complex carbohydrate, breaks off one of those glucose molecules so it can undergo cellular respiration. So you would have heard about plants. They undergo photosynthesis and they create glucose. They store glucose, unless, like for potatoes, for instance, they store it in the root. So when there is no sun and the plant continually needs glucose in order to undergo and create energy, it can tap into its starch resources to create energy or to redeem energy from those glucose molecules. Now, we don't form starch. We form our polysaccharides in the form of glycogen. And where do we store that glycogen? We store it in the liver and we store it in the muscle. So you'll learn in cellular respiration, when we have a muscle cell that needs to burn energy, it gets it specifically from the glycogen. It takes that glycogen, and if you were to look at these complex carbohydrates, they're literally strung together. It will just break one off, and now you've got a glucose molecule to use and create energy from it. We're going to undergo an exergonic reaction where we take that glucose molecule, again, this glucose molecule, and turn it into energy. So where do we store most of our energy? Again, don't necessarily need to know this per se, but 75% of all glycogen is stored in the muscle, and about 25% stored in the liver. And this will make much more sense when we talk about this on Thursday. You'll see the processes in which we generate glucose. And I'll have a lot of terms for you to review as we go through how we generate glucose and how we burn glucose. The last bit of complex carbohydrates is cellulose. And cellulose is very prominent in cell walls of plants. That's what gives those plants those very strong cellular structures, these walls on the outside that we can't necessarily digest. Other species, other animals can digest these, but we have no use for it. The only use that we use this for is the fiber that's in our diet. So if you ever look again at the back of the label, it says dietary fiber, or it says insoluble fiber. This is fiber that we can't digest, but it comprises most of the roughage that you find in feces. That's the stuff that's left over. So if you ever eat like a bunch of salads, you know you've got bulkier stools, as opposed to if you had a low fiber diet, you probably know what your stool is like. So that is our carbohydrates. One thing I want to mention too, because you saw me drawing our glucose molecules in a ring. What's not, what typically longer molecules like glucose, and let me just draw out that diagram I was drawing with 1, 2, 3, 4, 5, and then one of these. These are carbons attached at each single one of these points. Typically what happens with these long carbohydrate molecules is that they will start to form a ring. This one will attach back to this point over here, and then you get a ring-formed carbohydrate. And when you look at glucose that next Thursday, you'll see that ring form, and we'll talk about how it gets broken down. We won't get into the real molecular breakdown, but we will get to the gist of the conformational changes that occur to break down and release our form of energy from it. So that is our carbohydrates. Let's take a look at lipids. And lipids are important because these are the most diverse form of biomolecules. Why is it so diverse? You think lipids, fats are bad. No, not really. Fats are not bad. Back in, I think maybe like 90s, early 2000s, we had like I think it was not the Atkins diet, we had like low-fat diets. People used to think that fat is really bad for us. And so people eliminated fat from their diets, but they found also there was a whole host of side effects that people were having, this not mental dysfunction, but reduced function, like mental function, etc. Why? Because fats are important for us. We have 4 categories that these fall under, that fall under the category of lipids. We have fatty acids, triglycerides, phospholipids, and steroids. I'll talk about these in just a second. What exactly are lipids? Lipids are long strings of typically with hydrocarbons. And they're comprised of a bunch of carbons and hydrogens. Things you can see, like for instance, on our next slide over, you can see a whole string of carbons, hydrocarbons, etc. These typically in room temperature are solid. Any fats that in room temperature that are solid at room temperature are typically fats. These are like your saturated fats, like butter and like animal fats. Anything that are liquid at room temperature is typically oils. So you see fats and oils. There's plant oils, there's fish oils, you name it. Another thing about lipids is that they are nonpolar. And you remember what I said about nonpolar? They are nonpolar covalent bonds. So things like this, you don't have any areas that are electronegative or electropositive. Those electrons are shared very equally. So therefore, if there are no electrical or electronegativity or electropositivity, there's no point where water can come in and attach to. Hence, you've probably heard the term that oil and water don't mix. Even though water is a universal solvent, it doesn't seem to cling on to anywhere where fats are present. Hence why you'll see a separation. And that has a profound influence on how we can get certain fats to move through our circulatory system. If you were to look at fats, there are fats in your blood. How do we get fats dissolved in the blood? Stay tuned in our endocrine lecture where we talk about how that takes place. The categories, fatty acids, which we'll talk about. These are typically our monomers that we'll be looking at. Triglycerides are our long-term storage of fat. Phospholipids are something that I'll talk about. We have a slide that talks about this. Phospholipids are amphiphilic, meaning they're both polar and nonpolar. They exhibit both properties. Amphi meaning like amphibian, both. And then philic, like philia, like necrophilia, the love of. Steroids are another example that I use. Complex and important, because steroids, which we'll talk about, are very important in our endocrine lecture. So let's take a look at our first example, which we're going to skip to triglycerides. And triglycerides, you'll see on here, again, we don't really need to know about the biochemistry behind this, so you don't really have to remember the structures of these. You don't have to remember how many carbons there are. I want you guys to get the general gist of it, that this is how we store fats. So here are our fatty acid molecules. This one example is palmitic acid. And palmitic acid is a long hydrocarbon chain attached to a carboxyl group. Hence the term why we call it fatty acids. It's an acid because it contains a carboxyl group. Now, the other molecule on here is a glycerol molecule. It is a type of sugar alcohol. And it has 3 attachment points right here where we use C-hydroxyl groups. And in this process, in order to get that fatty acid to bind to that glycerol molecule, we're going to undergo dehydration synthesis. And you guys already mentioned it earlier, is the loss of water. And in this loss of water, we also have to invest energy in it. So this is an endergonic reaction in order to build that molecule. And I know I had a question here earlier to my left that talked about what type of molecules are you building. This is exactly what I'm talking about. I'm attaching this fatty acid to the glycerol molecule. That's a reaction that involves energy in order to create. And when we invest that energy, we're going to see that a water molecule is lost. Hence why we have dehydration synthesis. So this is what's called, and again, not a term you need to know, but it's called an ester linkage, where we have the hydrogen from a hydroxyl group attached to the hydroxyl group from a carboxylic acid, specifically this fatty acid. And you remove these, the H2O, and you get the water loss, and you get that ester linkage. We repeat that 3 times, and we have a triglyceride, okay, or otherwise known as a triacylglycerol. You don't really need to know that, but triglyceride is a fat. So if you see this, this is our way of storing fat for us, triglycerides. And if you were to think about breaking this molecule apart, if we're going to break this for energy, Imagine breaking bonds here, all these types of bonds. This is going to release a bunch of energy. If you don't believe me, look at fats, look at fuels, look at oils. You burn them and they release a lot of energy. Hence why if we need to store energy, you've had a huge caloric intake and you need to store that energy, you might store some of it as glycogen, but most of it where you're going to end up storing it is in the form of triglycerides, fat deposit, because we've got a lot of energy in those bonds. The next thing we're going to talk about is phospholipids. So I want to take that last concept that we talked about, that triglyceride, and I want to take one of those attachment points where that ester linkage is supposed to happen between the fatty acid and that third point on that glycerol molecule. And instead, I'm not going to attach a fatty acid to it. I'm going to attach a phosphate group. And this phosphate group, I'd like you guys to be familiar with. This phosphate group right here is a very electron-hungry functional group. Why? Because it has two things that I talked about, phosphorus and oxygen. Now you got a bunch of double bonded oxygen attached to that phosphorus. That is our phosphate group. And it really longs for electrons. So this makes it very electronegative. And on the other side, as I mentioned earlier, water doesn't have any attachment points. So if you were a water molecule, which side do you think you would be most attracted to? Would you be attracted to the area where you have all these nonpolar covalent bonds, or would you be attracted more to the phosphate group? Phosphate group. So here you see this molecule, the phospholipid, exhibits that property, that amphiphilic. Amphiphilic. You might even see the term amphipathic. Two terms are interchangeable, where you have a nonpolar side and a polar side. And this is one of, in my opinion, and probably many other opinions in the scientific community, is one of the most important molecules in our, if you believe in evolution, the evolution from unicellular organisms to complex cellular organisms. Because this molecule exhibits that amphiphilic property of having both a polar head and a nonpolar side. This, when you look at these molecules, we're going to look at this on Tuesday. When you dump a bunch of phospholipids into water, okay, and I'm just going to draw a bunch of polar heads like this. You dump enough of it in water, you're going to get the polar sides, and you'll see all my circles are the polar sides. And all the double fatty acid tails all are hydrophobic. They're going to hide away from water. You're going to get a bunch of water on the outside, and you're going to get all the fatty acid tails that are hydrophobic to gather on the inside. This, you don't need to know this term, it's called a micelle. It's one side where you have an aqueous environment, and on the inside you have the opposite of an aqueous environment is a anti-aqueous environment. Don't quote me on that. I don't know what the real term is. So that is what we have. And if you were to take an even bigger amount of phospholipids, right? If you were to take an even bigger phospholipid, what happens is you have, and you're going to have phospholipids that are going to create what's called a phospholipid bilayer. I'm just going to draw it like this so that you guys get the point. Where the inside is going to have an aqueous environment. Remember, these are the polar heads where you're going to have water on the inside. You have polar heads on the outside that are going to be exposed to the water on the outside. So water on the inside, water on the outside. And you're going to have this impermeable barrier. And I don't want to say impermeable because it's not impermeable. But you're going to have an area right here that's going to maintain separation from the internal environment and the external environment. This is going to be the major setup of what we're going to be talking about on Tuesday that's going to create the cell. The cellular environment where we're going to have an intracellular environment and an extracellular environment. And if you look at the creation of cells in the evolution of time, we found that, and this is some of the studies that were done by USC when they were looking at some of the early formed cells. They had like these dust particles that were able to create, and these dust particles had these complex lipids on them that created the separation of the aqueous environment that had toxic stuff in it, had sulfur compounds and all this stuff, and an inside environment that was separated from that could allow stuff to kind of move in and out between these phospholipids, that created this hospitable environment, something where a premature cell could survive on its own, where it keeps all the bad stuff out, keeps all the good stuff on the inside. So that is the whole concept between phospholipids. Let's take a look at our unsaturated fats and saturated fats. Again, not really that important. So I'm okay if you guys don't really get this, but this is really the difference between unsaturated fats and saturated fats. Don't forget, don't remember any of these trans, cis, cis. These are just the configuration, the molecular configuration of these. It's perfectly okay. But basically, saturated fats are they're linear in nature, and they can stack together and they can get compressed. So you get a lot of fats that are compressed together in a small amount of space. Hence why they can form these solid structures at room temperature. Unsaturated fats have a tendency to be kinked, meaning they are just kinky in nature, and they have a tendency to kind of just float along and never really stack on top of each other. So if you look ever at like peanut oils, you ever look at olive oils, for instance, they're kinked fats that just kind of float around. So they are not as high in caloric density. Steroids. I want to talk about. Steroids are the key component that I was talking about that we're going to be looking at in our endocrine lecture. And the main characteristic of steroids, if you were to look at a steroid, you can tell it's a steroid based on the molecular structure. And here you can see 4 fused rings of carbon rings attached to one another. You'll see some functional groups like a carbonyl group, you'll see a hydroxyl group attached at certain areas along that group. And wherever those functional groups attach will determine the characteristic of that particular steroid. We've got numerous types of steroids that we will be talking about a little bit. If you want to know which ones these are, this one's testosterone. The one on here is dexamethasone, which is a drug that's used for anti-inflammatory. The other one on here is cholesterol. Cholesterol is another important one. Again, you see 4 fused rings attached to a hydrocarbon on there. Something that you'll be able to see in our cellular physiology lecture. You're going to be seeing cholesterol intermixed with a bunch of phospholipids. That's going to give this fluidity. Again, stay tuned on Tuesday. We'll talk more about that. Again, so if you were to see these 4-ring compounds, you can tell they're steroids. And they serve as signaling molecules. If you're, as I mentioned earlier, we saw testosterone. Testosterone that's released by the testes has many targets that it can affect. It'll come in, and whenever testosterone cell binds to testosterone, it'll undergo a bunch of changes. It's our way of literally sending a chemical signal to a bunch of other cells. And that's all based, again, on those attachment points. Not something that I would really have you guys memorize per se, but what I'd like you guys to be familiar with is just the inherent structure of those. So what questions do we have about the fats? Shall we move on? Okay, I'll take that as yes. Proteins. Proteins, if you were to take a look at the monomer of proteins, the single unit of proteins, are those amino acids. So you take that CC, right? And this was our, we had H, and again, you don't need to know the structures of these. We took our ethane, we got rid of that one of the hydrogens, and we added our amine to it, our amine functional group. And in fact, I'm going to get rid of 2 of these, and I'm going to attach a double bond O to it. I have an amine group attached to a carboxylic group attached. That is our amine group, our amino acid. And it's an amino acid because of that, oh, there, I'm missing the other H. This makes it an acid, the carboxylic acid. So this amino acid is a very versatile amino acid because there's 20 forms that are available on this earth. We have 20 particular amino acids. You see this essential amino acid mnemonic? Yeah, don't worry about these. That's okay. I just want you to just be able to see the, and you can see this on the next slide over. Again, you don't have to memorize these amino acids. I just want to be able to show you that on here we have different characteristics of all these amino acids, of which we have 20. And the main difference between all of these, if you were to take a look at each of these structures right here, these are all similar. Repeating for each single one afterwards. The main difference is the functional group that attaches to that center carbon, that R with the big circle I drew around. This is the question of what attaches to that amino acid. That's going to define what type of amino acid it is. And you can see a bunch on here. You'll see glycine, alanine, serine, threonine, cysteine, valine, leucine. You go through all of these. Tyrosine, aspartic acid, glutamic acid. You'll see a bunch that we do produce. Of which, and again, I'm not going to have you guys really differentiate between the two. We have essential amino acids, which are required for us to be taken in through diet. We have non-essential amino acids. These are amino acids that we can produce in our human body. So if you think about why certain, when you look at diets and they say get foods that are rich in amino acids, make sure you have a varied diet, it's because you want to make sure that you're getting all of your essential amino acids. Yeah. So now what we're going to do is we're going to take each of those amino acids and we're going to string them together to create a protein. We're going to polymerize it to create a protein. And what we're going to do, let me just show you from our last slide over. Again, we would undergo dehydration synthesis. Again, something you don't need to know. But if I have here's my amine number 1, and then I'm going to have my amino acid number 2, where I have N, etc. And then I have my H. What I'm going to do is I'm going to take these two together. I'm going to remove that, and I'm going to form a bond between this carboxyl carbon and attach that to the other end. And what that's going to do, it's going to string 2 amino acids together. Now take that image and string a bunch of amino acids all together, like a long Pez string, or just like a pearl necklace of amino acids that are all strung together, and that's exactly what you're going to be able to see. One side is going to be the amino acid end, which you saw here. Here's the amine group. The other end is going to be the carboxylic acid end. That string that you're going to be able to see here, if I can sort of point that out. 1, 2, 3. You guys get the idea. This is going to string together and we're going to create a long protein. Now the thing is that you need to know about proteins. We don't have just one amino acid. We don't have just 2 amino acids. Proteins can be really, really, really big. And I mean, you can string amino acids. I'm talking about hundreds of thousands of amino acids that go on forever. And we make a bunch of proteins. What we're going to be talking about on Tuesday, I believe, we're going to talk about the manufacturing of proteins, how cells produce these proteins. Because these proteins vary in their functions, which we'll talk about in a little bit. What I want to get at first is the difference in the structures of them. And keep in mind that these structures, we start off with a primary structure, a secondary structure, a tertiary structure, and a quaternary structure. These structures build on top of each other. So you got to really just focus and just kind of think 3-4 dimensionally with me. So we take that primary structure and we string just some random, we'll take glycine, alanine, and serine. Glycine, alanine, and serine. We're just going to string a bunch together. That is our primary structure. So if you look at primary structure of a protein, it's literally just a sequence of amino acids strung together, all the way from the very beginning, all the way to the very end. Now take that, and what we're going to look at is the secondary structure. And the secondary structure is looking at the hydrogen bonds that form all along this primary structure. And maybe somewhere on, let's take a look at tyrosine. Oh, there's tyrosine. There's an OH right there. There's another, glutamic acid has another electronegative side on there. Some of these amino acids can now stick together due to hydrogen bonding. So what happens in the secondary structure, all along that primary structure, somewhere those functional groups that attach will be attracted by hydrogen bonding because of electronegativity and electropositivity. When they happen in repeated sequence, you get a series of structures, of which which we'll talk about. An alpha helix is a bunch of amino acids. Again, take that same structure that we looked at, string them all together, and now we get some sides that stick together through hydrogen bonding. And we see a repeated sequence that occurs. And you'll see that it creates a spiral. Let me just take a glance at some of these right here. Secondary structure, if you were to look at the protein being drawn, you can see the secondary structure forming because of these little hydrogen bonds that form. The other thing that you would typically see are pleated sheets, meaning you'll get the primary sequence that goes this way, and then it kind of wraps around and wraps back the other way, and it creates hydrogen bonds that form like rungs on a ladder. And it forms literally like a sheet, a pleated sheet on top of each other. And if you were to take a look at what that looks like, a pleated sheet looks exactly like this. And based on that structure, based on those sequences and the functional groups that attach to those sequences, again, you're going to get another structure. And because those hydrogen bonds are weak, get a bunch of them together and they hold together very strongly. So it's not uncommon when you look at a protein and you look down, you will see these structures. So that's the secondary structure. Again, based on hydrogen bonding. The third, again, now stay with me. Third is protein folding. And based on the regions of this protein that's starting to fold, there'll be some areas that will be hydrophobic and areas that will be hydrophilic. Some areas that will want to stay away from water and areas that will love water and get more exposed to it. There's also going to be these sulfide bridges. If I go back and take a look at any of these that have sulfide bridges, I should see one. Oh, look, methionine has got one. Cysteine has them. You get any of these two that are somewhat close together. And if they come close enough, and you might not see it on here, but if they get close enough, they'll bridge together and they'll hold very strongly. So that's the tertiary structure. Let me zoom in on this right here. So take a look at our tertiary structure. Again, if you were to follow the sequence of amino acids, that's your primary. Here you see the hydrogen bonds forming. That creates the alpha helices. Here's our beta pleated sheet. This is our secondary structure. And then these areas right here might be filled with a bunch of hydrophobic amino acids. Hydrophilic amino acids, where they want to get exposed to water. Maybe these areas on the inside are hydrophobic, so they want to stay away from water. So there's a lot that goes into protein folding. Let me get into the last one. I forgot this other one. The quaternary structure. All this one's a little bit simpler. You're taking protein strands that have folded together, and you get another protein strand that comes and folds together, and they'll kind of just stick together like a lock and key. And that's exactly what hemoglobin is. Hemoglobin is made from 4 different protein subunits, and I'm talking about 4 of these structures that stick together and create one functional protein. It's kind of like, is it Transformers? Do Transformers do that? They kind of bind together and create like one super. No, Transformers don't do that. Voltron. You guys remember Voltron? God, I'm so old.
Power Rangers.
Power Rangers. Power Rangers do that? Yeah, there you go. Power Rangers. So Power Rangers come together and they create some. That's exactly what they do. They form together and they have one function together. And Power Rangers, whatever they did, they were meant to kick ass. That's all they were supposed to do. That's basically what a protein does. A protein does specifically that, and they can be made from multiple structures. What questions do we have about these sequences? I know it's kind of a lot building up on top of each other. Let's take a look at these right here. One thing I want to bring up, is anybody familiar with the protein folding programs out there? There's There's a, I forgot what institution does it, but I think it was at MIT or one of the other big ones. They have these computers that will take sequences of primary structures. And if you ever look at DNA, and we'll talk about this on Tuesday, you look at DNA encodes for these proteins. So you take these proteins of the primary sequence and you create these long strings, and they can be hundreds of millions of amino acids strung together. The question is, how does that protein fold together, accounting for the secondary structures, the tertiary structures, and the quaternary structures? And there's a lot of computational effort that goes into folding this protein that will determine its inherent shape and determine its inherent function as a molecule. So we take that, and there's no single computer that can do it. So what this program does is, if you install it on your computer, what you're doing is you're donating the computational resources of your computer. It stays on all night, and it runs your processor, and it runs your computer quite hard. And you're contributing the efforts, and it's really cool. They string a bunch of computers together to create this huge supercomputer to calculate the protein folding that occurs. So you can enroll in the program. I don't think you get paid for it, but you contribute quite a bit to science because they've got to figure these things out. If they've mapped the human genome and they want to know ultimately how a protein folds, they have to use the computational efforts of multiple people. And I think as computers get stronger and stronger now, obviously they're making a lot of progress, and they can determine how it folds out. So that is the protein structure. Now that I've mentioned it, the ultimate shape of a protein will determine its function. And you can either have two types of protein shapes. Either it's globular, meaning like a globe, like this thing, or you can have a fibrous protein. Fibrous protein you might think of like beef jerky. When you tear them apart, you look at muscle proteins. Those are fibrous proteins. Think of connective, like tendons and ligaments happen to be fibrous. These are more like your connective tissue. These are more of like your globular proteins, things we'll talk about in just a bit. And if you take a look at the globular proteins on here, you can see the areas on here, and the areas in yellow are hydrophilic, meaning they want to be exposed to water. The areas in green have a tendency to hide away from water, so they're hydrophobic. So again, the tertiary structure depends on how this thing folds. You'll see things like an alpha helix right here, secondary structure. In our fibrous protein, you can see 3 different subunits. How do I know? I see 3 different colors. And again, you'll have hydrogen bonding that will attach these structures together to lend itself to the shape of the protein. So the shape of the protein, again, determines its function. Same with this other globular protein. Same thing. You can start to see where different subunits come together. Hydrophobic areas on the inside. Subunits coming together. And then it serves its function. So proteins are really the most diverse and most common type of proteins. The globular proteins we find in almost everything. If you ever do lab work, they're always looking at different proteins, and there's different proteins that they're looking at that will correspond to different functions. You ever look at liver enzymes? Enzymes are proteins. If you look at all the stuff in labs, we'll be able to look at labs and I'll be able to point that out later. Okay, so I kind of use the term enzymes, and that's exactly what I'm getting to, is that certain proteins can become enzymes, right? If you're ever to look at an enzyme, I would say 99% of the time, it's a protein. And I say that other 1% can actually have maybe made from RNA, but really the main idea is that all proteins are not enzymes. So if you were to use that example, enzymes are all proteins, but not all proteins are enzymes. So enzymes are specific. They're a protein with a function. It has one task only, and that's its, and what that task is inherently dependent on what that enzyme is. These enzymes have certain areas to them. If you look at the creation of this protein. We've talked about primary, secondary, tertiary, quaternary structures. There's certain areas based on the way it folds that attracts certain things such as our reactants. And those things that bind to it are called active sites. So if you were to see substrate is just a fancy word for things that bind to it. It's things that bind to something else. And so your substrates in this case would be man and woman that would bind to that enzyme. And that enzyme only binds to whatever it's tuned to bind to. Yes, I'm sorry. It's attracted to it. It literally will fit like a lock and key. So if you look at the shape of man, and this enzyme is supposed to bind to it, it'll be the exact opposite formation of it that allows it to literally bind together. So that's the active site. The enzymes are extremely efficient at what they do. They find a substrate, man binds to it. They find woman, binds to it. And they catalyze a reaction. And what I mean by that, stay tuned, I'll talk about that in a couple slides. We also have the ability to turn off enzymes on and off. If I wanted to do something, turn it on. If I wanted to stop doing it, turn it off. And there's areas on that enzyme that I can do to turn it on and turn it off. So the next thing on here is when we look at enzymes, there's a lot of enzymes. And you can see there's some enzymes on here, but this is not all of them. If you were to ever look at the list of enzymes, we have hundreds of millions of enzymes. Maybe that's an over-exaggeration, but we have lots of enzymes in the human body. And how do we name them? The naming convention is actually fairly simple. If you were to look at every enzyme, the name of that enzyme ends in ACE. So if you were to look at, let's go back to, who are our lactose intolerant people again? Okay, great. So we're going to use that example. Lactose is a combination of glucose and galactose. It's a disaccharide that's bound together that is that milk sugar. People that are lactose intolerant do not have lactase. Lactase is the enzyme that breaks apart lactose. So if you lack that enzyme, maybe you don't lack the enzyme, you might not produce enough of that enzyme to break it down. Therefore, it starts making its way through the gastrointestinal tract. The bacteria that's in our large intestine doesn't react to lactose very well, and it causes flatulence, or it causes stomach ache, or whatever it causes that are typical symptoms of people that are lactose intolerant. That lactase enzyme has one job, and that's to break down lactose into glucose and galactose. Every one of these enzymes have one job. Hydrolase. Its reaction, or what it's responsible is for hydrolysis. Use water to break apart a molecule. That's all it does. For instance, an example of a hydrolase, and these are the classes of enzymes. An example of lipase is to find out a lipid and break it down into a smaller lipid. That's really it. If you look at lactase, that was another form of a hydrolase. It was to turn lactose into glucose and galactose. The other one on there, the exact opposite of hydrolase, is a ligase. It's to join two or more chemicals together to undergo an anabolic reaction. The first one I talked about, hydrolase, was a catabolic reaction. So in this case, this allows for the synthesis. If I'm going to turn glucose into glycogen, I'm going to employ some form of a ligase or a polymerase. The other one that I want to look at that'll also be relevant for us is phosphorylase. Phosphorylase, the purpose of a phosphorylase enzyme is to catalyze the transfer of a phosphate ion. That's phosphate, when you see PI, it's phosphate ion, to a substrate. It means add a phosphate ion to that molecule. So once we do that, well, we'll talk about what phosphorylases are for. You'll be able to see that at the end of today's lecture. So like I said, just to recap, enzymes are specific to what they do, and they have active sites that bind to the substrates, and it only binds to that substrate. So like I said, go back to that initial slide that we talked about earlier when we talked about activation energy. And that activation energy that we talked about was that little push on that roller coaster. It was that match that we needed to set something on fire. It was that little bit of energy that you need to invest to continue that reaction to bring it to its final product. And to use that example earlier, I said man and woman, we need a little bit of activation energy. And in that activation energy, whatever it is to get them into the right position, to finally bump into each other and produce our product, our baby. Now, enzymes, think about what enzymes do. If they've got an active site, let's take let me use an example of a matchmaker. Matchmakers work really well. I'm sure amongst your friends you've got matchmakers that are like, hey, let me meet so-and-so. And they come up and they grab you and they grab the other person and they say, I want you to meet so-and-so. And then they get you two together and then you come together and then you form baby. That's exactly what those matchmakers do. They know the man, they know the woman, they bring them together to create the final product. So in those cases, enzymes reduce the amount of energy that's required. The product of man and woman or reactants stays the same. If we measure the amount of potential energy, that remains the same. If we measure the potential energy in baby, that remains the same. We're not changing the structure of baby. We're not changing the structure of man and woman. All we're doing is we're helping that process take place a little bit. They're matchmaking to bring them together. So in that case, an enzyme is programmed to attach to a man, attach to a woman, and help the reaction come together and form the product. Okay? They are meant exactly for that. So in that case, we reduce the activation energy. There's no change in the potential energy. So I'm going to draw that change in potential energy here, which we mentioned as negative delta G. There's no change in this. This remains the same. It's just the activation energy that comes in. So this again, to use another example, if you had a stack of papers, I couldn't even really think of this would be like a flamethrower. I can't even really use that as an example. You'll learn this about me. I'll try and play around with analogies and I'll just be like, no, that doesn't work, just kidding. In this case, that's where we reduce the amount of effort or energy that's required to start the reaction. If I were to take this whole beaker filled with reactants and products, and I throw, and let's say this reaction starts to slow down, right? Because I needed a good amount of energy to keep this going. If I threw some enzyme, if I threw a matchmaker in there, that one's going to come in and grab A and grab B together, put them together, and form more of the product. It speeds up the reaction because you use less energy. And that I'm referring to as activation energy. What questions do we have about that? Because I'm going to expand on this. Good? All right. So now we have to think about that enzyme. And what we're going to be looking at is ways to regulate the activity of that enzyme. Maybe that matchmaker works way too fast. Slow down. I don't want that much baby. I need more men and women. I don't need too much baby. In fact, you've created too many babies, turn that enzyme off. Okay? So in this case, what we're going to be looking at, competitive inhibition, we're going to be looking at allosteric regulation. And there's two different ways of looking at where we can compete with. Now think of the example of competitive inhibition that I was talking about earlier. I've got an enzyme that comes in. Here's our matchmaker, and we have the substrate, which is man, and we can use the example of woman later. Substrate comes in. This is one of our reactants that comes in, and it's supposed to bind to the active site on that enzyme. So this is our active site. A competitive inhibitor comes in and binds to the active site because the structure of the inhibitor mimics that of the substrate. So as I mentioned earlier, lock and key, this competitive inhibitor kind of looks like that key, and it can fit into that area. And what that ends up doing, it literally will block that active site. So think of that example that I mentioned earlier. You have that matchmaker that comes in and say, hey, I really want you to be so-and-so. You're going to come up to that man, but now you're going to get this competitive inhibitor that comes in here, and I've used this term before, comes in and just kind of cock blocks right there, right in the center. Just says, no, 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 don't do that, don't do that. In fact, I'm just going to get in the way. That's exactly what a competitive inhibitor does. Now, a reversible competitive inhibitor still competes for the active site, but it's reversible. Pretty self-explanatory. So it may bind sometimes, and in that case, if it does bind, it prevents a substrate from binding. But if we were to use this example earlier, a bunch of men, women, baby, and I threw that enzyme in there, the amount of competition that takes place is dependent on how much reversible competitive inhibitor I have. So if I toss in like one cock blocker in there, it's just going to block one enzyme. If I toss a bunch in there, it's going to come in and start competing. You got more of these competing for the active site than you do have your reactants. Does that make sense? So think about it this way. If I have more substrate is likely to bind to an enzyme than a reversible competitive inhibitor. If I add more reversible competitive inhibitor in there, you have more of these that compete than substrates. So that's how you can regulate, and that is something that we typically see of ways to compete for substrates, to modulate how much product we form, because it'll take that substrate, it'll undergo its conformational changes, and then it'll produce that final product. But get stuff that comes in and competes for that site, and you can limit the rate of reaction. Now, while we've been talking about the active site, in terms of competition for the active site, we have to talk about another site. And this is what's called the allosteric site. It's another site on the enzyme that is not the active site. So if this is your active site, there's an allosteric site that can bind to it. And in this case, this is an allosteric inhibitor. An allosteric inhibitor comes in, and when it binds to that allosteric site, it creates an entire change to the conformational change of that protein. These can hydrogen bind to this area and cause a change in the shape of the protein, or change the shape of the enzyme. And when you change the shape of the enzyme, you can change the shape of the active site. That's why proteins, when you look at proteins, any minor change, any tweaks to the protein can really cause dysfunction of that enzyme. It could potentially be that people that are lactose intolerant may have some mutation on there that changes the active site. It's now distorted so it can't bind to, so that lactose can't bind to that active site. You still have the enzyme, it's just misshapen. Okay. Give you a relevant example. Sickle cell anemia. People that have sickle cell anemia, the shape of the hemoglobin, actually it's the shape of the red blood cell, but the shape of the hemoglobin may not lend itself to be able to attach to oxygen. Again, that might make more sense when we go on later. But the main idea is attach an allosteric inhibitor and you distort the active site, substrate can't bind to it. Now the converse of that is an allosteric activation. Take that same allosteric site, attach an allosteric activator, and that now activates the enzyme site. So that active site can now bind to the substrate. So in other words, in order for this enzyme to operate, it needs its partner. It needs this allosteric activator that binds to the allosteric site to cause a conformational change to the active site that allows it to bind to the substrate. I know I strung a lot of words there, but that's basically the summary of it. So inhibitor and activator. Allosteric on the allosteric site. All right, so looking at the enzyme phosphorylation, we're going to take a look at, and I'm not really going to even go into this, but enzyme phosphorylation. There was a phosphorylase enzyme that comes in that phosphorylates that enzyme. Typically what you would see that would activate an enzyme is typically a phosphate ion. That's something that you'll commonly see. In order to turn on this enzyme, I need a phosphorylase to come in here and attach to that enzyme. And yes, I did use that example of an enzyme attaching to another enzyme. There's a lot of enzymatic reactions that require numerous enzymes to activate it. So it's like a matchmaker. Take that matchmaker enzyme, and that needs another enzyme to come in and turn on that matchmaker. You can string a bunch of enzymes together to create that one function. So you take that phosphate ion, attach it to that enzyme, and then it turns it on. Typically, you would see this. This is an example of an enzyme that's involved in cellular division, something that we'll talk about on Tuesday. Cellular division takes place, and there's something that will turn this back on. And they'll do it so that it can stop that cell from continually dividing. And we find that if there's any mutations to this enzyme, we may find that that enzyme might not turn off, or that enzyme might not be turned on anymore to stop cellular division from taking place. So any mutations to an enzyme can potentially cause a cell to continually divide if it affects specifically this enzyme. I don't want to lose you guys too much on that, so I'm going to skip this altogether. Any way you look at it, this is just a way of turning it on. Other ways to modulate an enzyme from taking place. Doesn't always require an inhibitor or an activator. Other things that we can talk about. Temperature. If I get an enzyme and I turn up the temperature a little bit, think of that matchmaker. Now it's working up fast because it's got a lot of kinetic energy from all the heat. Now that matchmaker works really fast. If I cool it down, if I bring the temperature down, we reduce the kinetic energy, and now that matchmaker moves very slowly. You modulate the rate of the reaction due to temperature. You want to get that sugar to dissolve fast in water, heat it up. If you want to slow it down, cool it down quite a bit. pH. If I start introducing hydrogen ions in the bath, hydrogen ions can come in and start binding to the enzyme. And that could start to denature some of the proteins. Think about this. When you eat beef jerky, or if you're tofu jerky, or whatever, anything protein you take in, when you're eating it, it goes into your stomach that's filled with acids. Part of that breaking down, you start to see the hydrogen ions that start to attack it and start to change the shape of the protein and denaturing it. That's a term that I'm talking about, denaturing a protein. By denaturing it, you start to render that protein to become ineffective. Okay? So take an enzyme, put it in an acidic bath, and that can start to change the effectiveness of that enzyme. Substrate concentration. Matchmaker will not work very well if there's not enough of your reactants in place. So if I have less reactants, those enzymes, they're just looking around trying to find man and woman. They can't find it. You start dumping a bunch of reactants in place, and now they start working faster. Again, Le Chatelier's principle. The rate of reaction depends on the rate of your reactants. Increase the number of these, you produce a lot more of these. Reduce the amount of these, and you produce less of your product. So with that in place, you now have the idea, and the last couple slides just I want to finish up with, is this lends itself to the whole negative feedback system. I want you guys to take what you learned on Tuesday in terms of the feedback system and apply it to this. Is that the rate slows as the product accumulates. Once, take that bath again earlier. Take that enzyme that's in there. If I'm trying to create a balance of, and I don't want too much man and woman, I don't want too much of baby, I want just the reaction to take place. I don't want to have it too fast or too slow. If it happens too fast, then I get too many babies that are produced, too many products that are produced. Less of my reactants. Therefore, the reaction slows down. Okay? If it's too slow, right? If it's too slow, I don't have enough baby, I have more of my reactants, man and woman, therefore the enzymatic reaction speeds up. And start introducing all the inhibitors and activators that can be introduced into the system to regulate the rate of how much we produce something. That is the whole system behind negative feedback. We want it to operate in a certain way, and when there's numerous physiological mechanisms that we introduce in the way to slow the rate down or speed it up if we need it to. The last thing I'm going to kind of brush through, which is still of utmost importance, are nucleic acids. I've got 1, 2, 3 slides left. Nucleic acids are the fourth macromolecule that I want to get into. And this is the structure of DNA and RNA. Something, again, we'll talk about on Monday that we'll get into some detail about, enough for you that you should be able to answer some questions on the quiz, and then real technically not really worry about it afterwards, because this goes into a lot more detail that I don't need you guys to get into. Again, a lot of this is biology. DNA is called deoxyribonucleic acid. RNA is called ribonucleic acid. So these are the structures, and as you know, DNA is the backbone for our basically human genome project. It's the sequence for what makes us who we are. All the components that we're ever made of is all stored in our DNA. And the way it's stored, if we take a look at the monomers are the nucleotides. These are nucleic acids that are formed. And we have, if we look at the monomer, the nucleic acid is comprised of 3 parts: a nitrogenous base, a pentose sugar, pentose meaning 5, and it's 5-carbon sugar, and a phosphate group. And you can see where these are. These green things, phosphate ions. Pentose sugar, take a look at that pentagram right there. That's our pentose sugar. This right here is our nitrogenous base. The nitrogenous base, we have 4 different nitrogenous bases. Actually, technically we have 5, but for DNA, we have 4. You don't need to know pyrimidines, you don't need to know purines, you don't need to know the names of these. Don't worry about that. We do have thymine and cytosine, adenine and guanine. These are single ring bases and double ring bases. At no point am I going to ask you which one. Thymine, is the thymine a single ring base or is guanine a double ring base? It's not really important to physiology. But the thing that we do need to understand is that adenine only binds to thymine. A double ring only binds to a single. Adenine to thymine and guanine to cytosine. There's no rules outside of that. They only bind to A to T and G to C. So if I were to take a look at this side of the sequence right here, A, G, T, C, and you'll see that the way DNA forms, phosphate, pentose, phosphate, pentose, phosphate, pentose, it strings together. And you have your nitrogenous bases attached together. I'm sorry, let me restate that. Nitrogenous bases attached to the pentose sugar. It creates this sequence of AGTC, AGTC, a bunch of nitrogenous bases that create this entire long sequence. Did you guys ever watch Jurassic Park? Or Jurassic Park, and they showed you, if you ever watched the portion where they're looking at the DNA sequence of dinosaurs, and it was a bunch of these ATGCAG, and it was like this really long string. It's exactly what it is. If you were to zoom in, if I were to zoom in on your DNA and look at the sequence for your eye color, I would see that it's going to be this hundreds of thousands, millions of gajillions of these nitrogenous bases that will give us the sequence of your DNA. These are genes that encode specifically for that. And if you were to look at what's called the complementary sequence, adenine, if I only had one side of it, adenine would bind to thymine, guanine would bind to cytosine, thymine would bind to adenine, cytosine would bind to guanine. So when we learn cellular division later on, you'll see how DNA will come in and it'll get spliced. The thing is about DNA, I kind of forgot to mention, DNA is a double helix. You probably have already seen this before. It forms this look to it. It's a double helix. And these right here, you would see the nitrogenous bases on the inside. And they're held together by hydrogen bonds. You see these two right here? These are the hydrogen bonds that form, that create this long ladder of DNA. And this area to this area could be the sequence for your hair color, could be the sequence for skin color. It gives you pretty much everything in there. There's all the different genes that we have that encode for these. This could encode for certain proteins. This could encode for certain enzymes, tons of it. So that ladder that's formed together is the hydrogenous bases. RNA, ribonucleic acid, is a single strand of nucleic acids. And we'll talk about that on Tuesday. The 3 types of RNA, again, I'm not going to really require this out of you guys, but RNA, we've got 3 types of RNA. One that's going to be partly involved, part of the construction of a ribosome, which we'll talk about Tuesday. tRNA is what's called an amino acid transfer. We go into this is actually one of your first lab assignments that we're going to end up doing is how do we sequence out the construction of a protein from DNA. We're going to go from DNA to RNA to the protein itself. Messenger RNA, again, we'll talk about on Tuesday. Now, take one specific nucleic acid, one of these DNA RNA structures. We're going to take one nucleic acid where we had a, this was specifically an adenosine, this was an adenine nitrogenous base, and we attach, here's our, there it is, adenine, this is our adenine. We attach that pentose sugar, which we call ribose, and instead of a single phosphate group that you saw earlier, that same green thing you saw here, instead of a single phosphate group, we string 3 phosphate groups together. The name of this structure is called adenosine triphosphate, and it's an important molecule because this forms the energy currency in our body. It is what holds a lot of energy in it. If you look at energy transfer, all of this is all contained within these phosphate groups. Each of these bonds of phosphate groups are very strong. They've got a lot of energy that's contained. And if you take this ATP molecule and we break off one of those phosphate ions, that's going to release energy. Remember, we're breaking this apart. It's a catabolic reaction. We take ATP, break off a phosphate group, and we get ADP, which adenine diphosphate, plus your phosphate ion, plus, don't forget, this is an exergonic reaction, energy. Tons of energy that can be coupled to endergonic reactions. Things such as building your protein. So if you're building that protein, you're going to be doing that in the lab assignment. It's a really easy lab assignment. If you're building a protein, it's going to require energy, and that energy comes in the form of ATP that's required. So with that, that breaks off our phosphate group. If you were to ever think about, has anybody ever watched The Matrix before? Yeah, you remember The Matrix? There was a group of, and again, I'm going to digress a little because I know it's like 9:40 and I know you guys are eager to go. In The Matrix, there was this a bunch of series of animations that were done before called the Animatrix, and they do the whole thing that talks about why the machines were taking humans and harnessing energy from them. There's a portion where Morpheus says the humans were nothing more than a battery, and he's talking about all that bioenergy that's available. Where does that bioenergy Where does that energy come from? Is it in the form of heat? No, it's actually in the form of ATP. So in the early in the Animatrix, they talked about the not ATP, but they talked about the energy that they were able to harness, and they are able. This is basically bioavailable energy. If you had a whole cup of ATP, that is energy that can be used to convert into readily form of energy. So.